Okay, let's kick things off. The really wild thing about the paper we're looking at today, it's this jellyfish protein.
Mm-hmm.
It makes other creatures glow green, like totally by itself. You don't need any other jellyfish bits or anything.
Yeah, and that was just mind-blowing back then.
Yeah.
Because before GFP, if you wanted to see genes at work in a living cell,
It was messy, right?
totally, the methods were often really harsh.
Mhm.
You know, they could damage the cells, even kill them sometimes,
Yeah,
which kind of defeats the purpose if you want to watch life happening.
exactly. So today we're diving deep into that original paper that just, well, cracked this whole thing open. The 1994 Science paper introducing Green Fluorescent Protein, GFP.
That's the one.
Yeah.
And we're also looking at some common frustrations people run into reading scientific papers, especially in biology. And there's this great analysis we found that really breaks down the impact of the GFP paper from like multiple angles.
Right. So our mission really is to get why this discovery was such a huge deal, why it completely changed biological research. And we'll try to do that keeping in mind, how tricky these papers can sometimes be to read.
Definitely. And just a little background context first.
Yeah, good idea.
Before GFP, tracking gene activity often meant using these reporter enzymes like beta-galactosidase.
Right.
And they usually needed some extra chemical added to create a color change, or you had to fix and kill the cells to see anything. Not ideal for watching things live.
So scientists were really hunting for something better, something gentler, more direct.
They were. And then you have this observation, this protein, GFP, from the jellyfish Aequorea victoria. It just glows in a test tube. No extra stuff needed.
Which sparks the big question.
The crucial question. Could you take this protein, put it in a different living thing, and would it still work? Could it glow there too?
Could this be the key? The way to finally watch cells live?
That curiosity was absolutely central. Could this weird jellyfish thing be a tool? And the paper starts answering that with, honestly, a pretty straightforward experiment.
Which brings us nicely to figure one. So what's happening here? The researchers took the GFP gene, the instructions for making it,
Yep, the DNA sequence from the jellyfish.
and they put it into E. coli,
Yeah.
the standard lab bacteria.
Exactly. They essentially gave these bacteria the recipe for GFP. Now imagine, because we can't see the figure, but imagine a petri dish under UV light.
Okay.
You've got your normal E. coli on one side, maybe,
Yeah.
and on the other, the ones that got the GFP gene.
And the result, bam.
Yeah, pretty much. The bacteria with the GFP gene, they glowed bright green under that blue or UV light. The regular ones, nothing.
So this is the first big punch. You can see gene expression, see the protein being made in a living organism without adding chemicals or killing it. Just shine a light.
It was huge. That visual proof was the first, really solid evidence that GFP could work in a totally different life form, a bacterium. And it didn't seem to need any special jellyfish-only helpers.
It suggested the glowing mechanism was sort of self-contained.
Pretty much, yeah. Which leads right into figure 2.
Yeah.
This one's a bit more technical, looking at light properties.
Right, spectra. Excitation and emission. What does that actually tell us?
Okay, so think of it like a light fingerprint. Excitation is the color of light the protein absorbs or gets excited by. Emission is the color it then gives off the glow.
And they compared the fingerprint of the GFP made in the bacteria?
To the fingerprint of the original GFP,
Yeah.
purified straight from the jellyfish. They were basically identical, like superimposable.
Okay, why is that so important?
Because it confirmed two critical things. One, the protein was folding up correctly inside the bacteria, getting its proper 3D shape.
Like protein origami.
Exactly. And two, the chromophore. That's the specific little bit inside the protein that actually makes the light it was forming correctly, too. All by itself, inside the bacteria.
So the bacteria weren't just like carrying around some pre-made glow stick from the jellyfish. They were building it from the instructions, and it worked perfectly.
Precisely. It proved the process was autonomous. It didn't need the jellyfish environment. Monumental, really.
Foundational. Okay, so bacteria. Check. What about something more complex?
That was the next logical step, and they didn't mess around. They went straight for an animal model.
Figure 3. The roundworm, C. elegans. What was the setup here?
Okay, so they put the GFP gene into the worm, but here's the clever part. They hook it up to the control switch, the promoter of a specific worm gene, MEX7.
And MEX7 is only active where?
In certain tetraceptor neurons. So they were basically telling the GFP gene, only turn on, only make the glowing protein in these specific cells, like wiring a light switch to just one room.
Okay, got it. And what did figure 3 actually look like? What did they see in the worm?
Well, if you were looking at the worm larva under the microscope, you'd see specific neurons glowing green. Not the whole worm, just those touch neurons where MEC7 is supposed to be active.
Whoa. So you could literally see individual specific cells light up inside a living animal.
For the first time, pretty much like that, yes. You could pinpoint where a gene was active in a multicellular organism in real time. Think about what that means for studying development or brain function.
It's incredible. So it works in eukaryotes too, not just bacteria. And you can use it to track where and when specific genes are turned on in particular cells within a living creature.
Exactly right. The pattern of glowing cells matched the known MEC7 pattern perfectly. And again, crucially, no extra chemicals needed for the worms to glow. It just worked.
Did they notice anything else? Like how stable was the glow?
Yeah, they noted it was generally stable, but it could fade a bit if you hit it with strong light for too long photo bleaching. But interestingly, they also saw it could sometimes recover, which hinted at, dynamic processes happening in the live worm.
So, okay. Figure 1 glows in bacteria. Figure 2 folds and forms correctly on its own. Figure 3 works in an animal, can target specific cells. You really start to see the power unlock across these figures.
Absolutely. It just systematically built the case. It laid the foundation for a whole new way of doing biology.
So let's shift gears a bit and talk about why this was so innovative, drawing on that analysis source we have. What really sparked this research?
Well, as we kind of touched on, there was this huge pressing need in the research community. Biologists desperately wanted a way to watch genes and proteins inside living cells without wrecking them.
The existing tools just weren't cutting it.
Not for live cell imaging, no. And then there was just pure scientific curiosity, wasn't there? Could this weird glowing protein from a jellyfish actually function all by itself somewhere else? It's a great what-if question.
That combination seems key, the practical need and the fundamental question, and the innovation itself. It wasn't some fancy new machine, was it?
No, not really. That's what's so elegant. It was more of a conceptual leap combined with a really direct and clever experimental plan.
Right, just clone the gene, stick it in bacteria, see what happens, then try it in a worm, compare the light properties, simple steps, profound outcome.
The real breakthrough was demonstrating that autonomous function, the folding, the chromophore formation, the light emission, could happen when you just transplanted the gene. That concept was revolutionary.
And the environment mattered too, didn't it? The collaborations.
Oh, definitely. This wasn't done in a vacuum. You had Martin Chalfie's lab, experts in C. elegans, who really needed this kind of tool.
And crucially, Douglas Prasher's lab had already cloned the gene?
Yes, and Prasher shared the clone freely. That openness, sharing materials before publication even, was absolutely vital. Chalfie heard Prasher talk about GFP and immediately saw the potential.
That sharing culture in science when it works is so powerful.
It really is. Plus, the timing was right. The early 90s saw huge advances in cloning tech, and model organisms like E. coli and C. elegans were well established. The pieces were sort of in place.
And you have to give credit to the individual insights, too. Chalfie recognizing the potential, the bold move to go straight for in vivo tests.
And the fact that the authors actually put the plasmid information in the paper, making it easy for others to replicate and build on it immediately, that accelerated everything.
It's amazing Prasher almost gave up on it due to funding, isn't it? Shows how fragile discovery can be sometimes.
Yeah, bit of luck, perseverance. It all plays a role.
So this idea of old molecule, new use, can you unpack that a bit more in the context of GFP?
Sure. GFP itself wasn't new. Scientists knew it existed in jellyfish for decades. The innovation wasn't discovering the molecule, it was realizing its potential as a general-purpose biological marker.
So finding a completely new application for something already known?
Exactly. And theoretically, it showed that this light-making part, the chromophore, could form spontaneously, which wasn't necessarily assumed before. But the technical impact was the big one.
Establishing it as a portable fluorescent tag you could stick onto other proteins or just express in cells across different species.
Right. That opened the floodgates. It wasn't just a tool, it became the foundation for an entire toolbox of fluorescent proteins.
All the different colors, the sensors, the optogenetic stuff.
Enhanced GFP, YFP, CFP, RFP, Sherry, FRT sensors, calcium indicators. It all snowballed from his initial discovery. Its influence is just immense across basic science, medicine, biotech, everywhere.
Deservedly Nobel Prize-winning work, then.
Oh, absolutely. The 2008 Nobel in Chemistry for Shimomura, Chalfie, and Cian recognized that transformative power.
So if you had to boil down the impact of this one paper into a single sentence.
Okay. It enabled scientists to directly watch molecules and processes inside living cells and organisms, fundamentally changing how we study life. something like that.
I think that captures it. It really did change everything.
It really did.
So wrapping up this deep dive, it's just undeniable how massive the impact of this glowing jellyfish protein has been. It completely reshaped biology.
And, going back to those pain points of reading papers, this original GFP paper, sure, it has its technical details, its specific jargon like any scientific paper.
It can still be dense in places.
Right. But the core finding is so elegant, so visual, a protein that just lights up inside living cells. That central idea cuts through a lot of the complexity. And the figures, as we discussed, they really do tell the story quite clearly.
Yeah, they make the breakthrough pretty accessible, even if you don't catch every single detail of the methods. The pictures or the idea of the pictures speak volumes.
Makes the core message stick.
It really does, and it leaves you wondering, doesn't it? This came from a jellyfish just doing its thing. How many other completely unexpected tools or insights are just waiting out there in the natural world?
Right. What other phenomena are happening that, if we just look at them in the right way or ask the right kind of what-if question?
Could totally revolutionize science all over again. A fascinating thought to end on.